the introduction. I hope you can hear me all. Better? Okay, good afternoon. So you just heard about the importance of the gut microbiota in relation to the feed efficiency of pigs. So I'm going now to talk about the host and particularly about the gut. It is clear that because of its high metabolic activity and all the processes that are going on in the gut, it needs a lot of energy and protein, and this therefore is classified as one important energy sink. And therefore, if there are differences in length, structure, or any kind of function, then it could be assumed that there would be or that the gut would contribute differently to the feed efficiency of the animal. There's already some evidence in the literature that the gut influences the feed efficiency, or let's say the residual feed intake in pigs. But of course, there's still some controversy out there regarding the digestive and absorptive functions, morphology, the effect regarding the barrier function, and also regarding the mucosal innate immune response. So in ECO-FCE, our hypothesis was now that a low RFI, meaning a good feed efficiency, is related to an enhanced digestive and absorptive capacity, and also with an enhanced intestinal barrier function and a reduced innate immune response. So our objective was then to investigate the differences in size, structure, and function of the gut of finishing pigs, and in particular we focused on the intestinal morphology, duodenal saccharide, disaccharide activity, and jejunal permeability. Coming from these data, we also investigated differences in relation at the gene expression level in relation to sugar, short-chain fatty acid transport, tight junction proteins, and some genes in relation to the innate immune response. You already heard the complex experimental setup that we had in this project. In my talk, I will focus on the pigs that were kept in Austria. We have already, of course, the results for the other locations, but to keep it easy today, this is just from the Austrian trial. So we basically followed the pigs between day 42 and 98 post-weaning and selected the most extreme animals. So we had in Austria 8 low and 8 high RFI pigs selected. The gut tissue sampling occurred from day 102 to 105 post-weaning. The sampling was spread over a few days because we conducted an oozing chamber experiment with intestinal tissue explants. So regarding the gut, what kind of measurements did we take? The easiest one was to measure the length and the weight. Then we used classical morphology measurements, of course, disaccharides at the brush border. Then we conducted an oozing chamber experiment, and last but not least, the candidate gene expression. So we focused on the duodenum and jejunum a bit more than on the ileum and the cecum. Regarding the lab analysis, you can see, and what you could also already understand from the objectives, we used tissue explants, meaning tissue that was still alive, used an organ, used functional proteins, and then went down to the gene expression level. And in the end, we ran an ANOVA using the fixed effects of the RFI. Because this is just a subset of the animals, let me show the differences in the RFI of our selected pigs. You can see here that the difference is approximately 2 kilograms. The pigs were also characterized by a trend for a difference in the average daily feed intake. So our high RFI pigs ate about half a kilo more than the low RFI pigs, whereas the growth performance was similar. It was about 1.1, 1.2 kg on average. I'm going to show now the most important results that we found for the Austrian pigs. We couldn't find a difference in the small intestinal length or in the cecal size. Also the weight was not different. We also determined other visceral organs which were not different in size, but we could detect some differences for the histomorphological measurements that we took. We measured classically the villus length, width and the crypt depth, and there were differences in the duodenum for the Austrian pigs. What we could see was a smaller crypt depth for the low RFI pigs and a trend for a broader villus width coming along with the low feed efficiency, with the low RFI value and the high feed efficiency. We also counted goblet cells and intraepithelial lymphocytes. We couldn't detect a difference for the lymphocytes, but for the goblet cells, basically in the duodenum, our low RFI pigs had more goblet cells, whereas there was a trend for the jejunum and a significant difference for the cecum that low RFI pigs had lower number of goblet cells per villus length and crypt depth. Because goblet cell number and mucus production are very closely linked. You have the question? The question? So it's working again. Thank you. The question that arises right now is, so what could have caused these differences? Is this just in relation to the feed intake, because the pigs with high RFI ate more, or could it be also in relation to the microbiota, because it's known that the microbiota can stimulate mucus production? Regarding the villus width, this could give an indication of a greater villus surface area, which might be in relation to a greater digestive activity going on at brush border level and a greater absorption. And this is why we actually decided to analyze the disaccharidosis in the duodenum. What is interesting is that it's not the maltase that responded or was different for the different feed deficiencies, but that this was the lactase activity that showed the biggest difference. Remember that we're finishing pigs at the time of slaughter, so lactase activity should not have been so important, and if we go through our dietary composition, milk components were not really the essential ones anymore. The second disaccharidase that responded or was different for low and high RFI was the sucrase. Also maybe not so important, but no effect, as I said, for the maltase. The question here is now, does the greater lactase activity really have an importance for the RFI gain? And also what we cannot completely distinguish is this really host related, or does it actually show an impact of the gut microbiota? So let's move on to the Ussing chamber results. For those of you who are not really familiar with this technique, this is a technique that helps to determine differences in the gut barrier function, in the gut permeability, because the jejunum is a very long part of the small intestine. we agreed in this project to take our samples at about 2.5 meters cranial to the iliocecal junction. We could not find differences in electrophysiological parameters which give you an indication about ion flux and the tissue permeability. However, to differentiate better the paracellular from the transcellular flux, we also used molecular weight markers, one small molecular weight marker and one large molecular weight marker, and what we could see there was that our low RFI peaks had a higher permeability in comparison to the high RFI peaks. The question again that arises, yes, was this just related to the feed intake, or is this really a difference by the host in relation to the feed efficiency? Last but not least, we decided for a number of candidate genes to look at in relation to the innate immune response and also in relation to an interaction with the gut microbiota. Then we looked for transporters, glucose transporter and a monocarboxylide transporter, and also for tight junction proteins. Regarding the innate immune response genes, we saw differences for TNF-alpha and trends for the toll-like receptor 4 and the alkaline phosphatase. To guide you a little bit through it here, the toll-like receptor binds, for example, lipopolysaccharides, and in the signal transduction pathway there's also an upregulation of TNF-alpha with this, and also the alkaline phosphatase can detoxify certain metabolites from microbes. Therefore, there could be a link between luminal lipopolysaccharide processing and the mucosal response. For the other genes, unfortunately, there was no difference, so we couldn't detect here the same for the tight junction proteins as we could show for the intestinal permeability. So to sum this up, we could show differences for the duodenum and the distal jejunum, and cecum as well. For the goblet cells, we found some structural differences and some functional differences, and could partly also show differences at gene expression level. But of course, or what's often said and written in the literature, that the gene expression level does not completely correspond to the functional protein level. The effects that we could establish in relation to the RFID when linked to the innate immune response, and including the jejunal barrier function, which links us back to the intestinal microbiota, and possibly also in relation to digestion and host nutrient absorption, but still it needs to be clarified if this is just in relation to the feed intake level or and the intestinal nutrient flow. And now to make the whole story more complicated, we of course have also location effects, as we could see for the gut microbiota. And with this, I would like to thank you, the colleagues that were involved, and thank you for your attention.